Well, thank you, Professor Liu, for the introduction, and thank you, President Gore and Provost Ellis, for the honor of being a honorary doctorate. It was quite special, and I thank the university for that distinction. And upon this occasion, I found out that my fellow Caltech student is your provost. He was 1 year ahead of me, so it's quite a place to have a reunion. I have recently given this talk a number of times. This is a general audience, and I have perhaps I should have given a more introductory presentation on HIV in general. But as a research scientist, I typically like to talk about what we're doing now rather than what we did some years ago. My group, having worked on fundamental biology of HIV, HIV pathogenesis, and HIV therapy, for the past 10, 12 years, my colleagues and I have largely focused on sort of technical methods of preventing HIV infection. You all know you could prevent HIV infection by educating people, and that knowledge will help protect people. Or one could use, in the case of sexual transmission, condoms and other methods to prevent transmission. But the ultimate goal is to come up with a vaccine. And as I will tell you, vaccine has not been forthcoming up to this point. And therefore, we really need to continue to work on vaccine as well as, in parallel, work on other methods that could have a positive impact on transmission. So this is why, this is what we're doing, and I like to share with the audience, maybe not every technical detail of what we're doing, but the essence of what we're trying to do. So just a very few introductory remarks about the epidemic and the virus. I've said to a number of people, to me, this is one of the worst plagues in human history. It involves the entire globe. And already 35 million people have died from HIV infection, and another 35 million are living with the virus. And I think everyone here knows that other than a small minor percent, just about 99 to 99.5% of the infected individuals will die from HIV infection if untreated. So it's a deadly disease, but therapeutic advances were made. The epidemic was recognized in 81. By 1983, the virus was identified, and 84, it was proven to be the causative agent. And a blood test was developed in 85, and the first drug emerged in 87. And a series of drugs emerged thereafter, and the combination therapy that we helped develop were generally applied since 1996. So this automatic death sentence now has become a more manageable disease. And HIV cannot be cured, but it could be controlled. And a person infected could live pretty much a normal lifespan we expect, if treated. And of course, you know that particularly in sub-Saharan Africa, many of the infected individuals do not have access to therapy. But because of the good treatment in North America, in Europe, in Japan, Hong Kong, and to a lesser extent in mainland China, people are no longer dying acutely from this infection. And therefore, the awareness level has decreased among the population. And yet, each year, there are still more than 2 million new infections. So we all we all hear about, threatening diseases like SARS, flu, and now Ebola gaining attention, yet the numbers killed by those infections are dwarfed by HIV. And I like to say HIV is in some ways a silent tsunami that goes on every single day. And you could see with this kind of infection number, you have to work on prevention. That's the only way we're going to curtail the spread of this virus. But I just said we don't have a vaccine despite 25 plus years of research. And there's a complex explanation for that, but for the sake of time, I will simplify it. To me, there are two fundamental problems in coming up with a vaccine against HIV, and they both related to the fact that we have not been able to develop an antibody response to the virus that's very effective in neutralizing the virus. For most infections, if you're infected after a few weeks, after a few days or a few weeks, you mount an antibody response, most of that antibody response is going to be effective in neutralizing the virus. Not so for HIV. And it has to do with the fact that HIV, here, shown schematically, it's as you could see, is a virus that's wrapped in lipid bilayer. So it's an envelope virus with the central core and the RNA, 2 strands of RNA as genome. And on the surface are these glycoproteins, which are then blown up down here. We don't have the complete structure of the glycoprotein three-dimensionally, but we have many components. For example, the inner core structure has been solved for some years now, but the inner core is devoid of some of the variable sequences which are mimicked here. So I said variable sequences. That suggests that virus HIV strains differ from one from another. And those sequence variability could be substantially greater than what you find in influenza viruses. And for influenza viruses, we have to make a vaccine each year to tailor it to the ones that threaten to cause a human outbreak. And so you could see the variability of the virus is one major obstacle. Then on top of the variable protein sequences, there are these elements that further decorate the surface. And those are the sugars, the glycosylation, the glycans that are added on to the protein. And in fact, we know that half of the molecular mass of this spike, this protein spike at the surface, comprised of sugar. And these further shield the critical elements in the core of the protein that need to bind to the receptor and mediate entry of the virus into its target cell. So you could see that for example, this yellow bit might be a part that an antibody could target. But this yellow bit is well shielded by variable sequences as well glycan. And these pose formidable problems for us to induce a good virus neutralizing antibody response. That said, well, for those of you who follow the literature in this area, you would say, but wait a minute, I've been reading about all these magical antibodies, monoclonal antibodies in particular, in Nature, in Science, and in some of the very top journals. And that would be true. For example, this is the glycoprotein now turned upside down. And there are a series of antibodies that have been described in the literature that bind, say, the transmembrane GP41 down here, or at the tip, such as antibodies PG9, PGT145. Don't be bothered by these technical terms. Just think about it conceptually. And then there are antibodies that target this region, and antibodies that target these regions in part target the sugar, the glycan on the protein. And then an antibody to this region by such names, in fact, targets the very site that the receptor for the virus would bind to. So these antibodies, in fact, exist, and they could be pulled out, and they could neutralize the virus, and some with good potency, and others with good breadth. So that breadth meaning covering a rather large array of divergent viruses. So, but remember, these are antibodies that are pulled out by special cloning technology. They may not represent dominant antibody species that you will find in the blood of an infected individual. So when you go back, so this is sort of looking at things at very detailed level. But when you take a step back and say, okay, these antibodies exist, but how often do you find them in infected people? And it turns out that only about 10 to 20% of the infected individuals would make any kind of antibodies like these that would have sufficient potency and sufficient breadth. So it's rare, first of all. Among all the infected individuals, you find it in a small minority. Now, the other thing is when you analyze the antibody sequences, the genes of these antibodies, particularly focusing on the region that needs to bind to the epitope. Those regions are typically called the CDRs. So they're the variable sequences within the antibody. If this is well known to you, I apologize, but these CDRs, when you examine these sequences, they're extremely variable. Some of them are extremely long. And that's the process that we call affinity maturation. So our antibody responses, when we see a new bug, a new virus, will make an antibody. But this antibody will continue to be refined over time through a process we call affinity maturation. And in fact, all these magical antibodies have gone through extensive affinity maturation for years and years. So it's only in a small minority, and it takes years of antigenic stimulation to develop these antibody responses. Now, if you think about this, that tells you how tough the challenge is to come up with a vaccine, right? It takes lots of viruses to induce in a small percentage of people the right response, but after only many years. How are you going to recapitulate that with a vaccine shot or a few shots of a vaccine? It's exceedingly difficult. So this is, to me, the principal reason why we don't have a vaccine. But we do know what the problems are. And now many groups armed with this information are trying out new approaches to HIV vaccine development, including folks in my own group. Unless there's some magical event, I don't think that vaccine is going to come out in the next 5 to 10 years. So it's still another long journey before we get there. And in the meantime, the epidemic continues to spread. So we got to come up with alternative approaches, and that's what I wish to concentrate on today.